Well, dear students, in the previous class we had discussed about interspecific hybridization, about mule and Hindi we have discussed, similar examples I had given. After that topic, in the animal breeding, we have special techniques of breeding that we also called as, we also refer it as controlled breeding experiments, or we can say as controlled breeding techniques. In these techniques, right, these are the artificial techniques, yeah? controlled breeding experiments, or we can say techniques, because they are done artificially in the laboratories, hence they can be called as techniques also. There are different techniques to improve the breeds. We have seen hybridization, we have seen interspecific hybridization, we have seen inbreeding, we have seen outbreeding, we have seen outcrossing, we have seen. So there are many natural techniques for that. So apart from that, as the science developed, as the need also developed, so the scientists, dietitians, dairy technology when improved, when all this happened, many artificial techniques they started developing as far as to develop the more milk yielding breeds or more meat yielding bulls. Right? So all this was developed by three different techniques. I want to tell you one thing. Since I am using headphones, mic, you people don't use earphones while listening the class. You can listen the class just like that. Increase the volume in your mobile and listen to the class. You don't require the headphones to listen to the class. If you do it, there is a lot of echo. This is what few students have told. Echo is there. Since I am using headphones, you don't use your earphones or headphones to listen. I hope it's clear. Now, in this technique, what do you mean by controlled breeding techniques? The techniques that are used to manipulate, to increase to produce the new breeds for the more production of milk as far as cow, buffaloes are concerned, and for meat production as far as bulls are concerned. So there are few techniques, right, by which we can improve their yield, we can improve their production of milk, the amount of milk in dairy technology. That's what you study in the same thing of same chapter as White Revolution, brought about by Dr. Varghese Curie. Now, one question I will ask you. You might have seen in our exotic breeds we have discussed, Holstein, Friesian, New Jersey we have discussed. So now you might have observed, or the question should come to your mind. How come only these cows are there, more number? For example, in Holstein, Friesian, most of them are cows in the market, in the people's in the houses, in the farmers' houses, in the livestock who are maintaining. In the dairy technology, only cows are there. Rarely you see, very rarely you see the bull of HF, Holstein, Friesian, the bull of Jersey, right? So how these only cows are being produced? How these only dairy animals are being produced in a huge number? This question should have come to your mind, because you come across, you see only cows in the farmer's house or in the livestock or in the dairy technology and the dairy farms also. You come across so many cows, but you don't see bulls of these breeds, exotic breeds, the breeds particularly that are used for dairy purposes, right? You may not have seen at all the males of these breeds. So question should have arisen in your mind. So for that we have answered today in this topic. So what are the controlled breeding techniques? The techniques that are used to increase the production, increase the yield, and to produce better breeds by using artificial techniques. So such experiments and techniques are known as controlled breeding experiments or techniques. Among them, we have three we have discussed. One is known as artificial insemination. This is one technique, what we usually call as AI, usually done in cattle, right? Usually done in cattle, cows, buffaloes, and other livestock, right? This is done. What we call it as artificial insemination. The first one. The second one we have discussed multiple ovulation, and embryo transfer. This is one technique, right? We call this one as MOET, right? MOET, multiple ovulation and embryo transfer. It is also referred as SOET. What do you mean by S here? Super ovulation. it is known as superovulation. This heading remains the same. SOET or MOET. This will be asked for 2 marks or 3 marks in your final exam. Since it is a little deep topic, we will discuss in the next session. Today we will be discussing artificial insemination, and then the third technique in this topic today is known as sperm sexing. What it is known as? Sperm sexing. Right? So these are the 3 techniques that are mainly used in order to produce the desired breeds. Only cows, only desired cows, right? Clear. So among them, the first technique what we have is known as artificial insemination. As the name suggests, fine. Artificial. Here, the semen is quite natural. Semen is quite natural. Only the technique is artificial. The technique is artificial. Hence, it is known as artificial insemination. This insemination. What do you mean by insemination? Since this has already might have come in your human reproduction chapter, but that is yet to be dealt in online classes. What is insemination means? The process of transferring semen into female reproductive tract is what is known as insemination. What is the process of transferring or the process of deposition of semen into female reproductive tract is known as insemination. Now what is semen? It is a reproductive fluid, right? Reproductive fluid which has sperms in them, which has sperms. What are sperms? The male gametes, clear. So in that fluid, that liquid, that white colored, milky, thick, gummy, kind viscous, heavy fluid is what is known as semen, right? So in this semen, millions of sperms are suspended. So such a fluid is what is known as semen, right? The process of deposition of semen into female reproductive tract is what is known as insemination. Now the word, why do we use here artificial? Because this is done by artificial method in the laboratories, in the veterinary hospitals, in the hospitals, in the laboratories, in vitro condition, outside condition, fine. We add this, fine. We do it. So hence it is known as artificial insemination. By using syringes, semen is collected in this, right? What is happening here? In this process, the semen from the selected bull, which bull? Elite bull or selected bull, or the breed which you are using it for the purpose of crossbreeding to produce. Fine. Semen is collected from the selected bull. It is collected. How it is collected? By stimulation to that bull. Semen is released, ejaculation, the process of releasing of the semen from the male reproductive tract or male reproductive organ is what is known as ejaculation. Right? So by stimulation, by artificial means this semen is removed from the bull, ejaculated from the bull. That semen is collected. After collecting, it is stored from -79 to -196 degrees Celsius in a low temperature, very low temperature. Such a process of the technique of storing of semen or DNA or RNA or genes or pollen grains or cells, tissues, anything which are stored in so low temperature, -196, by using liquid nitrogen. By using what? Liquid nitrogen. By using liquid nitrogen, this semen is stored. Such a process is known as cryopreservation. Here in Greek, cryo refers to low temperature, pyro refers to high temperature. You might have seen in 8th chapter Human Health and Disease, I have told you antipyretic, against high temperature. The tablets that are used to reduce the temperature or to cure the fever, what is known as antipyretic. Pyro refers to high temperature, cryo refers to low temperature. So here this semen is stored in -196 degrees Celsius in order to retain its viable conditions, in order to retain its living properties. They are stored in a very low temperature by using liquid nitrogen. You know, nitrogen is a gas. When it is compressed to a low temperature, it becomes liquid, like you might have seen LPG, liquid petroleum gas, right? So similarly here, by using this nitrogen gas, this semen is stored. Such a process is what is known as cryopreservation. This cryopreservation is basically a general technique, not only for artificial insemination or not only for semen. As I already told you, this technique is used also to store genes, to store the DNA, RNA, fine, pollen grains, cells, tissues, fine, in plant tissue culture, animal tissue culture. So it's a general technique basically. Same technique is applied here, so that the semen from the bull is collected by stimulation process, ejaculation process. It is collected, then stored in the liquid nitrogen, such a process known as cryopreservation. So whenever it is required, it is taken into syringes, then, or it is known as catheter or syringes. Then the doctor will the gloves, right? Then this injection is inserted into female reproductive tract of cows, right? In the cattle, in the cows, in the buffaloes, in other livestock animals, right? Clear? This injection is injected into the female reproductive tract during the heat period. You may have come across what do you mean by heat period. This is a common technology, right? I mean common word. It is also used by our farmers, our own farmers. In villages also, right? Heat period is used. Terminology heat is used. So what do you mean by that? At that time it requires the semen or the sperms for fertilization, because it is sexually very active, right? Ovulation has taken place. Such a period is known as heat period. This terminology you would have come across in first chapter also, when you are studying the differences between estrous cycle and menstrual cycle. So during heat period, whenever they are fertile, whenever they have released their ova, so at that specific time, at that particular time, fine, this semen is injected into the female reproductive tract. So what is the advantage here? Advantage here is fertilization is 100% sure, right? 100% sure. Clear? So what is the method? Semen is collected, stored whenever it is required. During the heat period of the cow, it is taken to artificial syringes. Then from the syringes, it is injected into female reproductive tract. By this method, fine, we can produce many animals by using this technique, because naturally a bull can fertilize. Naturally, I am telling now, so that you can understand the benefit of this technique. Naturally, bull can fertilize around 500 to 600 cows when it naturally has sexual intercourse, right? Or copulation with the cows. In a year, the capacity of a bull, right, is to fertilize 500 to 600 cows. Clear? Understand? So whereas if you do it by artificial methods, right, semen is collected, only around 0.2 ml or 0.5 ml of semen is taken from the cryopreservation technique, from this, and that semen, very small quantity of semen, right, is used to inject the female. So by this, with the same amount of the semen, what is ejaculated by a bull in 1 year, which can normally, under natural conditions, it can fertilize up to 500 to 600 cows. Here we can fertilize more than 50,000 cows. Why? Because the quantity what we are using is very low, very small quantity, very specific, at the time of heat period only, right? And very less quantity, and this less quantity also used to have thousands of sperms. So by using this technique, we can fertilize more than 50,000 cows per year. So one more advantage is transportation of, right? This is a technique. In this, after that, it is a normal gestation period of cows. Gestation period of cows takes place. Then normal delivery takes place. Now let's talk about the advantages of artificial insemination. No need to maintain bull farms or to maintain the bulls. Only one bull is enough to fertilize more than 50,000 cows. No need to maintain bulls. Many bulls are not required here. Then transportation of animals from one place to other, whether it is cow or a bull. Transportation from one place to other places is not required, because the semen is present in the hospitals, in the veterinary hospitals with the veterinary doctors, right? So transportation of bulls can be avoided, right? wastage of semen can be avoided, then we can prevent STDs, sexually transmitted diseases, or the diseases that occurs or transmitted through genital tract. Fine, that also can be prevented. It also can be prevented because the natural condition when sexual reproduction takes place, when the copulation takes place between the bull and the cow, right here. So there are chances of transferring, infecting many sexual disorders or diseases that can be avoided because it is done under aseptic conditions, fine, by using syringes, aseptic syringes, sterile syringes, fine, sterile blouses, fine. A veterinary doctor uses, fine, catheter inserts into the female reproductive tract, fine, through the vagina. Then it is injected into the very close to the ovum. So here the fertilization is very much confirmed, and moreover it is injected into during the heat period of the cows. So this is the technique or the procedure how to do artificial insemination. Then the advantage are no need to maintain bull herds, transportation is avoided, genital disorders or STDs are avoided. Then many thousands of cows can be fertilized by the semen collected from one bull in a year. Fine. Normally, right? It will fertilize only around 500 to 600 bulls. Normally, clear. Whereas, for example, to understand this concept, for one ejaculation, the semen, the amount of the semen collected from one bull is used to inseminate. Artificial insemination is used to inseminate more than 70 cows per ejaculation. So by that you can calculate. So normally, naturally, in 1 year, in natural condition, the bull which can fertilize 500 to 600 cows, the same amount of semen is taken in vials, preserved, used in a very less quantity, so that we can fertilize more than 50,000 cows. So here many advantages are there. Sexually transmitted disease can be avoided. Transportation of animals from one place to another place for the purpose of fertilization is avoided. And during the heat period, it is injected. Artificial insemination is done. So here fertilization is very much confirmed. Clear? And by this you can use one bull to fertilize more than 50,000 cows. One selected bull, elite bull, selected bull, the good breed bull, right, can be selected, and one bull can be maintained to fertilize more than 50,000 cows. Right? This is the process, and the advantages. In this topic, I have given the introduction to controlled breeding experiments. What are they? Why are they? Definitions to what is insemination. Definition to the cryopreservation. These definitions can be asked for 1 mark also in your final exam. Fine. Clear? Do watch this class repeatedly. Today I thought of completing one more third topic, that is sperm sexing. Fine. In one more class we'll do MOAT, multiple ovulation embryo transfer. Very important topic. Fine. It has been asked many a times in final exams. Clear? The technique, name the technique or explain the procedure of increasing the herd of the animals. Fine. Students get confused here. In my next topic, I'll discuss multiple ovulation embryo transfer. Do take very care of yourself. Fine. Keep watching the classes. Fine. Make your own notes. Go through the study material. I hope it's clear. Fine. Don't use your headphones. The echo will be caused. Since I am using phones, fine. Don't use it. So do take care of yourself. God bless you. Keep studying. God bless you. Good luck.